Hi, this is M. Prasanna. Today we are discussing about the pyrimidine metabolism. So this pyrimidine metabolism, so this is heterocyclic ring and C4, C5 and C6 and N1 these are formed by the aspartic acid, and N3 and C2 this is formed by the carbomyl phosphate. So this is the source of C and N this thing. So pyrimidine is the first synthesis and it is later attached in ribose 5 phosphate. So biosynthesis of pyrimidine ribonucleotide. The synthesis of pyrimidines is a much simpler process and compared to that purines. So aspartate, glutamate, carbon dioxide contributes at two atoms and the formations of pyrimidine ring. The pyrimidine ring is first synthesized and attached to the ribose phosphate and is in contrast of purine nucleotide synthesis where the purine ring is built upon the pre-existing ribose 5-phosphate. The formation of carbomyl phosphate. The carbomyl phosphate is formed by the ATP and glutamine and carbon dioxide. So reactions catalyzed by the CPS carbomyl phosphate 2 and carbon dioxide combined with the glutamate and combines with the ATP. The formations of carbomyl phosphate. The difference between carbomyl phosphate 1 and carbomyl phosphate 2. The site will be mitochondria. Carbomyl phosphate 1 is mitochondria and here this 2 is cytoplasm. This pathway is urea and this pathway is pyrimidine and also effect of N-acetyl glutamate, and here no effect, and source of ammonia and glutamate, inhibitors of CTP. So what are the condensation? The condensation carbomyl phosphate condense with the aspartate and form the carbomyl aspartate and catalyzed by the aspartate transcarbomylase, carbomyl phosphate and aspartic acid to carbomyl aspartic acid. So ring closure, this occur via the loss of water. The reaction catalyzed by the dihydroorotase forming the dihydroorotic acid and carbomyl aspartic acid, dihydroorotic acid. So dehydration dehydrogenation is like a removal of hydrogen atoms to C5 and C6 by the dehydroorotase mitochondrial. So dihydroorotic acid form the orotic acid. So this orotic acid form the orotic monophosphate by the involving of orotic phosphoribosyl transferase. So this orotic monophosphate conversions of uric monophosphate. So the UMP to UDP, UDP to UTP formation by the ATP conversions of ADP releases. The formations of UTP to CTP by the involving of CTP synthesis by the involving of glutamate. So here the reductions of nucleotide diphosphate and their corresponding NADPs and ribonucleoside diphosphate is conversion of 2-dihydroxy ribonucleoside diphosphate. So oxidized to reduce by involving of thioreductase. So NADPH is conversions of NADP. So UDP conversions of UTP, UTP conversions of CTP by the CTP synthase, and UTP is conversion of dihydroxyuric phosphate by the DUMP by the of H2O and inorganic phosphate will be released. So some of the methylation of UMP. So DUMP D2 to TMP conversion. So by the thymidylation, very important and crucial step here. D is the uridine monophosphate to convert to thymidine monophosphate. This by the involving of thymidylate synthase enzymes. So solvase pathway. Pyrimidine like purines can also serve as a precursors of the solvase pathway to be converted to the respective nucleotides, and these reactions catalyzed by the pyrimidine phosphoribosyl transferase, which utilizes the PRPP as the source of ribose 5-phosphate. The solvase pathway pyrimidine base PR phosphoribosyl pyrophosphate, a conversion of pyrimidine nucleotides by the inorganic phosphate by the involving of pyrimidine phosphoribosyl transferase. So regulations of pyrimidine synthesis by carbomyl phosphate 2 aspartate carbomylase and dehydroorotase are present in multi-enzyme complex. So Orotate phosphoribosyl transferase and orotyl monophosphate like decarboxylase are present in single functional enzyme. Due to the clustering of the enzymes, the synthesis is well coordinated. Dehydroorotate dehydrogenase is mitochondrial enzyme. So carbomyl phosphate 2 and aspartate transcarbomylase and Orotate phosphoribosyl transferase and Orotate monophosphorylase decarboxylase are sensitive to allosteric regulations. Carbomyl phosphate 2 is main regulator enzymes in mammalian cells. like carbonyl phosphate 2 inhibited by the UTP and activated by the PRPP. Aspartate transcarbomylase, main regulations of the regulatory enzyme in the prokaryotics, inhibited by the CTP, activated by the ATP. Requirements of ATP for CTP synthesis and stimulatory effect of GTP and on CTP synthesis ensure a balanced synthesis of purines and pyrimidines. So degradations of pyrimidine nucleotides, and the pyrimidine nucleotides undergoes to similar reactions, dephosphorylation, deaminations, and cleavage of glycosidic bond like that of purine nucleotides to liberated liberated the nitrogenous, the cytosine, uracil, and thiamide. The bases are then degraded highly soluble product and beta alanine and beta amino isobutyric, and these are the amino acid which undergoes the transaminations and other reactions to finally produce the acetyl CoA and succinyl CoA. So how the cytosine to uracil to dihydroxyurate to beta alanine, and thiamide to dihydroxythiamine, and beta urate and beta amino isobutyric acid, the catabolism of purines. So disorders of pyrimidines, the orotic aciduria and orotic aciduria 1 and orotic aciduria 2 only by the decarboxylase enzymes. So what are the features? How lack of feedback inhibition like orotic acid production, excessive UMP inhibitors of OMP decarboxylation, and rapidly growing of the cell effect anemia, retardation growth, also crystal like excreted in urine like causing of urinary obstruction. Both responded to urine as it is converted to UTP. This act as a feedback inhibitor. So this how deficiency in liver mitochondrial and transcarbomylase like linked undergoes to utilize the substrate like a carbomyl phosphate enters and inside stimulates the pyrimidine nucleotide biosynthesis and leading to cause of orotic aciduria. So how allopurinol, the drugs may be participated like precipitated like orotic aciduria. Allopurinol is one of the drugs. So this resulting inhibits the orotyl monophosphate decarboxylase. So Rice syndrome, this is the consideration as a secondary orotic acid aciduria. So it is believed that a defect in ornithine transcarbomylase or urea cycle causes the accumulations of carbomyl phosphate. This is then diverted to form the increases the synthesis of excretion and orotic acid. Thank you. Today we are going to discuss about the nucleotide like in pyrimidine metabolism. Thank you very much.